Hey biology students, I'm so glad that you're tuning in to this Weinberg tutorial because I know that this is one of the more complex topics and very complex. There's lots of things that you need to understand about genetics in order to be able to do the Weinberg problems. And so first we're going to go over the equations that you need to use. So the equations are, whoops, forgot to get my pencil. So P plus Q equals 1 and P squared plus 2PQ plus Q squared equals 1. So those are the two equations we're going to use. And then you also need to know what the P and the Q stand for. So P always represents the dominant allele. Okay, so that could be, for example, capital T. Q always represents the recessive allele. Right? And so if they're asking you to find the frequency of the dominant or recessive allele, you're looking for P or Q. Now, if they're asking you for the frequency of a genotype, then you're looking for, if they're asking you for the homozygous dominant, then they're asking you for P squared, right? That would be like big T, big T. Or the homozygous recessive would be Q squared, and that would be like little t, little t. Or the heterozygous, and that's going to be 2PQ. So if they're asking you for the frequency of an allele, this is what you're going to, so this is the allele frequencies, okay? And then this would be what you're calculating if they're asking you for a genotypic frequency. All right? Okay, so let's go ahead and get that in for Q. Whoops. If we plug 0.4, right? With our first example, it says the allele for allele T for the ability to taste a particular chemical is dominant over allele little t for the inability to taste that chemical. So right away we know that big T is going to represent our dominant allele. And that's also going to represent P. And then the recessive allele is little t. And that represents Q. Okay? So it says 400 university students were surveyed and 64 were found to be non-tasters. So if they can't taste, that means that they have to be homozygous recessive. So that we know that TT would be our homozygous recessive genotype. And that also equals Q squared, which up here it tells us that there's 64, right? And it's 64 out of 400. And so 64 out of 400, oops, is going to be 0.16. And so if they were asking us for the genotypic frequency of homozygous recessive individuals, we would say, well, that's 16% or 0.16. However, they want us to calculate the percentage of heterozygous individuals. So let's go ahead and find T, because once we find T, then we can put it into this equation, P plus Q equals 1, right? And we can find P. So we need to find T. So remember that T equals Q, right? And so we need to take Q squared. We know that that's equal to 0.16, but if we square both sides, right, Q equals 0.4. And now we can plug this 0.4, right, in for Q, and we can solve for P. And so we have P equals 1 minus Q. And if we plug that 4 in for Q, it would be P equals 1 minus 0.4, which is going to give us P equals 0.6, right? Now we have, we know P and we know Q. And in order to find the heterozygous individuals, remember from the last slide, 2PQ is equal to the heterozygous. So the heterozygous frequency is going to equal 2PQ, and then we just plug in. So 2 times 0.6 times 0.4. And what is that? Right, 0.48. And so 0.48 is the frequency of our heterozygous students, or the percentage of our heterozygous students, right? And that's our answer. Okay, let's move on to the next example. It says, in corn, purple kernels are dominant to yellow. So right away, I always like to write this down, because then I know what they're giving me in the problem. So let's use a capital P for the dominant allele. You know what? We better not, because P is also in our equilibrium. So let's use C. So C equals purple, and little c equals yellow, right? Because purple is dominant to yellow. It says a random sample of 100 kernels is taken from a population in Hardy-Weinberg equilibrium, and it's found that 9 kernels are yellow and 91 are purple. Well, so what do we use? Do we use to find our answer? Because it's asking for the frequency of the yellow allele, right? And some students want to start with the 91 kernels that are purple, but remember, those 91 purple kernels, they can be CC, big C, or big C, little c, right? They can be either homozygous dominant or heterozygous. Both of those genotypes are going to give us purple seeds. Only the homozygous recessive genotype is going to give us the yellow color, right? And so we know that 9 out of 100 are going to be homozygous recessive, or little c, little c. And if we put that into decimal form, that's 0.09, right? And that's going to give us what? Right, that's going to give us Q squared, because Q squared is equal to our homozygous recessive genotypic frequency. Now, so we have Q squared equals 0.09. This gives us the genotypic frequency, right? This is equal to CC, but we need to know what the frequency of the yellow allele, not the yellow genotype. So we need to do the same thing we did on the other problem. We need to square both sides, and now we have Q equal to 0.3. And this is our answer. This is our frequency of the yellow allele. All right. Well, I hope this has helped you, and if you have any questions, you can email me or message me in Canvas. All right?